One other than me.
Yeah, cool. Hi guys, I'm Liz. Okay. So we're part of this campus ministry called Alpha Omega, and tomorrow evening we're showing a screening of the film Blood Brother. I don't know if you guys have heard of it, but it's basically about this kid who he moves to India and he spends his whole life serving and helping these HIV/AIDS orphans in India. So we're having it tomorrow.
Tomorrow at 6 p.m. Thursday at the Social Sciences in the big auditorium, and it's a really cool movie. It has won Sundance Awards and things like that. So you guys should check it out if you're free.
Free snacks?
You can bring snacks.
That's the way it real. Virology related, I might add.
Nope.
A couple of other announcements. Our Science Talk speaker yesterday, at least one of you went to Science Talk, was from Georgia Tech and gave a hell of a talk. It was something you missed. You're worse off for it. We'll never know how worse. He actually has just funded or gotten funded and participates in an REU. So as I push those of you who want to go into research to applying for these things, he gave a specific plug to this. So his name is Manu Black, as you saw in my previous plugs. And there's they have what does EPIC stand for? Engineering Biological Machines. So it's a research experience for undergraduates that is a collaboration between MIT, Georgia Tech, and the University of Illinois. That's at Urbana-Champaign, not Chicago. So probably the least glamorous of the three locations. But their deadline is coming on February 14 or 15. He specifically encouraged applications from KSU. So I would encourage you to take a look. I put a link to the announcement. You can probably Google EPICS REU and get there just as quickly. But I put a link to it on the Facebook page. So again, I encourage you to think about applying to this and know that if you apply this, perhaps strangely, Kennesaw State has some brand name recognition with the people who run this program. He was very impressed with us. He said he was. Announcement number one. Number two, at least one person has already noticed that, and you can look on Facebook for this. So this is one of those ways to be a really lazy college professor. I'm going to award LCA points to people who write potential exam questions. So the idea is you guys get on the Facebook group and you think of what might make a good exam question, you ask it, and then your peers may answer it. So 4 points for writing what I judge to be a quality, coherent question. I don't know what my standard for that is, we'll have to see what we get. And then 4 points to the first person who answers it. You can write 2 questions, you can answer 2 questions for up to 16 points. I would encourage you to do it even beyond the LCA points, because one, it's a decent exercise, because you should think, what am I going to think is a good question? And that might lead you to writing a good question. You can get information, this is the whole idea of crowdsourcing, you can get ideas on what other people think is important. And a lot of other people think something is important, chances are it may be important, or it might convince me that it's important. I promise that, assuming I get some coherent questions, and I think Andrew has already given us some, I'll use something from this on the test. Maybe one question, maybe more. So it's a way to also write your own exam. Plus I have to do less work. So I would point out Andrew has already written a couple of questions. Ooh, and somebody already answered since last time I checked. By the way, Andrew, congratulations on your relationship. I had Andrew post some time ago, but it was his most recent post. It was quite fetching. Anyway, so there it is. You got to do this. I mean, there's a deadline before the test, but I'd like to see some activity, and then that allows me to look your questions. And then lastly, I'll keep pitching this throughout the semester. This Friday at noon, one of the biochemistry professors, Ella Mumaw, is going to give a talk that's essential. Well, it's on her particular enzymes. For some reason, she's interested in an enzyme that's in tree fungus. But she's doing bioinformatics now, so it's going to be a good talk about using computational power to look at enzyme activity. She also takes students for research here, so it's a good opportunity for you to expand that. And then next week is a famous guy from Cal Berkeley who will be in to do some genetics talks. But Friday at noon, I put it So we ended last time with double-stranded DNA, or DNA replication, which unsurprisingly looks a lot like replication in non-viral systems. And so we just, some DNA viruses will replicate in ways that are consistent with the replication fork schematic in the synthesis of the new strand is always 5' to 3', but in double-stranded DNA, the synthesis going in the opposite direction is the lagging strand, and the synthesis going with the fork is the leading strand. But in a circular DNA molecule, some double-stranded DNA viruses are circular, some are linear but made circular for replication. You have some global structural considerations, and that's a closed circle being replicated will form two circles, and as replication occurs, you get structures that look like that. That's known as theta replication, because the Greek letter theta. Fraternity and sorority people, forgive my Greek letter writing, it's not what it should be. And then an alternative structure that is sometimes seen is caused by a nick in the DNA, so that's a break in one strand of the two, followed by the replication fork moving around, or moving two replication forks in theta. There's one in sigma, but that it looks a lot like rolling circle replication that you see in some bacteria. That's known as sigma because of the Greek letter sigma. So structurally, they look like Greek letters. And again, like all of these topics we've been covering, we'll see these in some specificity later in the course. So then we will worry about double-stranded RNA replication, which unlike DNA replication, like DNA replication, can either be semi-conservative or conservative, which is to me, counterintuitive. So we have a double-stranded nucleic acid. The replication of which is dictated by the sequence of the parental strand, and so with straight analogous to replication, we get daughter molecules represented by the open boxes compared to parental molecules that are solid. Semi-conservative means each of the daughters has one strand of the parent. There are viruses, example, Rioviridae shown there, that the parental strands rejoin with one another and the daughter strands are entirely new molecules. How might one show that experimentally? You can't just look at an electron microscope and tell.
Tag.
How would you tag it?
The phosphate sugar pattern.
Okay, so what would you tag it with?
Fluorescent phosphorus.
Fluorescent phosphorus.
Radioactive.
Radioactive. Not easy. So a fluorophore is going to, practically speaking, mess things up at this level. But yes, you don't even need to do radioactive. The classic experiment was done with not the phosphate backbone, but the nitrogen, the nitrogen space. You can label it with N15, which is a stable isotope of nitrogen, and it's actually, as Spinal Tap would say, one heavier. So you can grow your virus and host in a medium that contains heavy nitrogen. It's a naturally occurring isotope. You can get pure sources of it, and the virus will incorporate nitrogen into its RNA, and then all you have to do is switch the media and look at the daughter molecules and how they separate in a centrifuge. The N15 labeled would be heavier than the N14 labeled, and these, of course, would be the same density. Like double-strand DNA, they also have a helicase, an enzyme that unwinds the double-strand. The single-stranded RNA viruses we have viruses that go from RNA to RNA. Oops. It has nothing to do with this slide. For which we need an RNA-dependent RNA polymerase. As opposed to the DNA viruses, this is not something hosts do. They don't replicate RNA from RNA. Thus, this is going to be virally encoded. It could be packaged in the capsid of the virus as well, so it could just be delivered along with the virus. It will also, of course, be encoded in the virus. And then the only other operative point here is plus strand synthesis of the negative strand is going to be associated with membrane-bound enzymes. More curious fact than anything else. There's nothing about nucleic acids, at least to me, there's nothing about nucleic acids that replication structure that requires presence of lipids. It's a curious factor of plus-strand RNAs that to replicate their genomes, they require a membrane-bound structure. So lipids are also required, and that's because some of the proteins are membrane anchored. This is not true minus. Then the other class of viruses we need to be worried about replication are the retroviruses. Of course, these all go from RNA to DNA. Different things can happen. When they're repackaged for exit, we go back to RNA. And so this is reverse transcription. We're going not from DNA to RNA, but from RNA to DNA, which is carried out by an enzyme, not coincidentally called reverse transcriptase. Reverse transcriptase also catalyzes the synthesis. So if we're a plus strand virus, then the plus is always relative to the mRNA. It encodes the protein. The minus strand is synthesized by reverse transcriptase. The complement, the plus strand DNA, is also synthesized by reverse transcriptase. So this is, from an enzymologist's point of view, a very weird thing. It's catalyzing a different reaction, that's different types of substrates rather than ribonucleotides. This activity uses deoxyribonucleotides, so it's a promiscuous enzyme. It's a multifunctional enzyme. I said the first or second day, one of the things about viral proteins is you often see multiple enzyme functions within the same polypeptide. Reverse transcriptase is our first example of that. That's chapter 8. I don't know how to make replication any more complex than that, at least it doesn't, so we'll move on. Remembering our handy mnemonic device, ATK. Attachment, entry, transcription, translation, genome replication, assembly, and exit. Like handy. Moving into chapter 8. By the way, I also forgot everything else. I think I said this last time, but I did as you're going out the door. Monday will be chapter 9. I can't imagine spending the whole class doing it, and I'll also talk about how I'm going to test it. I'm not exactly clear on it. I'll try to bring in some sort of sample exam. I'll try to have something to post, but this being my first time to the class, that means I basically have to write 2 exams. Laziness, I don't want to do, but I'll try to at least have some sample, like this is the sort of question I ask things for you. We'll talk about it on Monday, and I'll also try to put something up so you can see, just to get a feel for how I'm going to test you. I don't want, well, so I don't mind if you break down crying in the test because you didn't do well, as long as it's an accurate measurement of your knowledge. I want everybody to, of course, be happy to skip me down flowered pathway because they did awesome. But I want to test an accurate reflection of your knowledge. I don't want you to be surprised by anything. So I'll also take questions of, do you consider this important, do you consider that important? And generally the answer's gonna be, yes, it's important, because it was either in the text or we covered it in class, but I can maybe give you a little guidance on what I'm thinking about the degree of importance then. So I would like you to know what's coming on the test, so you know what to know. Baseline answers everything, but we can get a little more specific. So there's a carve time out for that Monday. In any event, so viral particles have to be stable and unstable at the same time. We're going to see an example of this in the paper we're going to talk about in a few minutes. But there's an equipoise between stability and instability. Particles themselves have to be stable to survive the environment. They have to be unstable in order to not survive in the host environment, because the genome has to get out in order for the cycle to continue. So with assembly and exit, we're looking at a juxtaposition between stability and instability. Your book talks about there being a trigger, such as a pH shift, which certainly happen. The story can be simply less stable over time, means you're unstable. So there could be a trigger, there can also just be things happening. Remember, we have helical and icosahedral structured viruses. The helical viruses are generally going to coat their nucleic acid in proteins, whereas that's the extent. We'll talk about it now. Nucleic acid's coated in proteins and then exit from the cell either by budding or by injury to the cell by an outside event, such as an animal eating the cell. Icosahedrals start out as protein shells known as procapsids. And then those procapsids have to be loaded with the genome and undergo maturation. Which is quite often an event, this is e.g. such as hydrolysis of peptide bonds or cleavage by proteases. So the scheme from your textbook is, for example, a procapsid. This is merely a protein shell consisting of coat proteins. It's in some precursor structure, hence the circularity. Genome is loaded. This is often an active process, but not necessarily always. It can be a happenstance, random chance event, and it could merely be the case that there are enough capsids in genomes that some get packaged, and that's effective enough to get the next generation of viruses out. So we're going to loading, and then this is the maturation from circular to icosahedral, involving, it could be post-translational modification, could be cleavage. Proteolysis is a post-translational modification. And then just know the existence of, at this point, scaffolding proteins. So we know viruses are small and their genomes are small. It's often a very tight squeeze, and that without other intervening molecules, packaging a genome in a virus would be a difficult thing. For example, DNA viruses, the single-strand DNA virus is going to have to pack a lot of negative charges because of that phosphate backbone into a small area. So you see nucleic acids are shown as these long stringy polymers because that's exactly what they are. If you want to pack them up, you have to worry about the repulsive electrostatic charges of the backbone. And so putting a nucleic acid into a small space is a thermodynamically unfavorable process that's often mediated by proteins that are not present in the final virus, known as scaffolding proteins. So they would ameliorate negative electrostatic repulsion by electrostatic attraction, so basic proteins, lots of arginines and lysines to mitigate the charge-to-charge the unfavorable charge-to-charge interactions. Envelope viruses have an envelope. I don't know how I'm going to touch on this, right? Question number 6. What do envelope viruses have that non-envelope viruses don't?
That'd be a good one.
I like that kind of one.
I think it'd be like the first question on the test.
Yeah, there's this tacit agreement, if I give you all A's, you give me good reviews.
Exactly.
I like that.
Would you want to do that?
Hell yeah, sounds good to me.
And I'd rather get into med school, re-scientist, and succeed.
We would with that A.
I'm sorry?
We would with that A.
Exactly.
That is the truth. They don't look at
There's also
GPA.
the reality of the MCAT. 4.0s with 18s on the MCAT are definite red flags.
What's going on? Just take it. You don't know.
I don't think there's a lot of virology.
Exactly, so.
Okay, they can either steal a membrane from the host, by, for example, budding off of the cell membranes. You have a mature but unenveloped capsid. Viral proteins, as we saw last time, that go integral membrane viral proteins that go via the endoplasmic reticulum Golgi apparatus to the cell surface are going to cluster because of either favorable interactions or an accumulation of interactions with the viral coat proteins. You see these are here. So we have coat proteins binding to viral proteins in the membrane. This is also an assembly process that can result in budding off of the virus and then the mature envelope virus. They can also cause de novo synthesis of a membrane, in which case you have an envelope virus still inside the cell. So how does it get out? Either by exocytosis or the much more dramatic lysis. I'm sure we'll see later in the semester pictures of poor cells chock full of bacteria that then explode, in which case the virus, if the cell bursts, then the virus is by definition exits of the cell. The same way that alien exits. You know Alien. It's 1978. I didn't even know Alien when it came out. Anyway. Alien exit the gastrointestinal system. And then we see a cutaway in your text of the viral membrane protein, again, very often glycosylated. That's an international symbol for a sugar group. So we have a transmembrane domain in the protein. That's why it had to be trafficked through the ER and the Golgi apparatus. And then an internal domain that binds either directly to the capsid protein, so this is a high affinity interaction, would have a low KD, or there's another protein that would be in the capsid coat, the inner space between the capsid and the coat that would mediate binding. There are two binding events here, one matrix protein, membrane protein, matrix protein, capsid protein. It's vanilla Rocky Road. Okay, that's replication, assembly, and exit, at least at any level we need to know them now. Let's talk about something interesting instead. How many people have ever read in detail a primary research paper? Pretty good. It's about a third of 10. I got 10. I got 10 and a couple of shakes, which I'll take as no's. I mean, she looked at it, but she didn't really read it. How many people really tried to read this one? even fewer. Okay. This may be less useful discussion than I would like. I want you to start doing it. So I will come up with something off of this paper for the test. And I'll take future papers and put them on the test as well. How many people listened to the quiz where they talked about this paper? So you made it through the discussions of the weather?
Yeah, it's like 6 minutes, like for a half hour.
They're gonna stop. They brought it up again in this
Okay.
week's.
So I want to, one, go through the science of the paper, and we can see some of these occurrences that, about which we've talked in a real research paper, and then how they might affect our understanding of the biology of herpes virus infections of foreskins. It's all an excuse for me to talk about foreskins. Okay, so today I want to not only have a discussion with you, but also sort of give the basics of how do you read a scientific paper. There's several different levels you can read a scientific paper. What's the most cursory one? You read the abstract, just believe whatever they say. What do you do next?
Discussion.
Well, that's several layers down. So yes, we want to actually read the paper. Because that way we can examine their evidence and know in detail, or form a detailed opinion about whether we think they're right, and what their data and conclusions mean for our understanding of the biology of what it is they're talking about. And then we can just look at the figures. So that's sort of intermediate level one. And so I just wanted to, well, start out with the abstract, and then I'll give you a few cues as to how we look at the paper, and then we'll bring up the data. The idea, at least for me, is we're tying not only a research paper about a subject in which we're interested into some of the replication, attachment, infection replication issues, but also we're bringing in those experimental methods that we were earlier in the course in a real sense. So I always like to, one, read the abstract, but two, look at their conclusion. Their results define a pathway for nuclear innate sensing of herpes simplex virus DNA by IFI-16 in infected HFF, and document a mechanism by which a virus can block this nuclear innate response. So the thing that'll happen to you immediately when you get into any field is you'll get buried in a sea of alphabet soup. So let's make ourselves a little key here. Okay, so HSV, that's easy enough. That's the herpes virus. HFF, human foreskin fibroblasts. What is a fibroblast?
It's like a cell that produces connective tissue.
You know the right answer the first time. Right. Cell that produces connective tissue. So we're talking about cells that are, well, let me ask this. What does the suffix blast mean? It was a big party, right? That was blast. Yeah, good. So what are cells that form things called?
Blast.
Yeah, okay, what's another word? What does blast connote? That it's a stem cell. So these are cells that are on their way to becoming foreskin. So these are targets for herpes virus. It also, I'm sure, is the case. I don't know this field, by the way. I had to weed through this alphabet soup just as much as you did. These are probably being used. Why would you use? The question answers yourself. Why would you use human foreskin fibroblasts as your model?
Because that's where the virus is infected and
It looks like natural targets of herpes, but what's another reason?
The cells are still in early development.
Yeah, it's black.
And so they will do what?
Mutate.
They will grow. What's the problem with most mammalian cells? They die. Gosh, I can't make this any more difficult. Right? When you grow cells in culture, so there are two basic types of cells. There are immortal cells, and there are, so immortal cells are things like cancer cells. They'll continue growing and growing, and you can divide them as many times as you like. And then there are primary cells, which are normal cells that will develop and stop growing and reach what's called senescence. Stem cells are a nice intermediate because they're much more like cells that are senescent, but they're not senescent yet, so they'll grow. So it's almost surely being used here because they're a good model system for targets of herpes virus, and herpes does not grow in all cells. And two, they're easy to work with in the lab, and that they're going to grow and they're not going to drop dead on you the way some other cells would. And a subsidiary point to that is cancer cells are weird. Like they've got genes turned on when they shouldn't be turned on and turned off when they shouldn't be turned off, and they make really good things to grow in lab, but may not be appropriate to reflect the biology of what you want to study. Okay, and then, where are we here? INF is interferon. What's an interferon?
It's a signaling molecule.
What does it signal?
It's
Help, I'm infected!
Immune system, yeah.
So it's a group of cells called cytokines that are going to signal either nearby cells or far away cells, depends on the type of cytokines they are, that there's an infection. So for our purposes, well, not our purposes, but in the paper, interferon, and when interferon is turned on and off, that's a signal that the cell has been infected. So it's a recognition of infection, and it's a response to infection. So production of interferon, I believe interferon beta is the one they look at here. I'll see if I'm right in a minute. Production of interferon equals one of the responses to infection. So interferon is turned, the gene is turned on, the protein is made when the cell recognizes the virus is in there. So IFI 16, and the title of the paper, it's biology, it's not that hard. Interferon inducible protein number 16, which may mean it's the 16th protein
There's others.
anyone ever looked at in response to interferon inducibility. It may be something else. I don't know the genesis of the term. It is a DNA, it's a DNA binding protein. So it's, as we'll see in a second,
Transcription factor.
turned on, it binds the viral DNA. So it's sort of a secondary response to infection. Interferon's on, interferon turns on the inducible protein. And then ICP 0, I guess it's the 0th protein. It's a viral protein. Ooh, let me do color. So it's a herpes protein, and this is it's a ubiquitin ligase. Not talked about in the paper very much, if at all. What does a ubiquitin ligase do? Does anybody know what ubiquitin is?
Ubiquitous means it's
No. Ubiquitin. Has anybody ever heard of ubiquitin? All right,
I don't know.
Travis?
ubiquitous means like universal.
So ubiquitous means something that's
Found
everywhere.
everywhere,
My reputation as an excellent teacher is ubiquitous. So what do you think ubiquitin is?
It's usable by all.
It's a post-translational modification. It's a protein that is added to other proteins everywhere. So it's found there. So it's named because it's found in multiple cellular compartments, so it's ubiquitous. It's ubiquitous. So if a protein has had a ubiquitin added to it, it has been ubiquitinated. So, this isn't funny. It's really, so ubiquitin is everywhere. Ubiquitin ligate put together ubiquitin onto target proteins. This is a mechanism for how the cell gets rid of proteins. So if a protein, for example, loses its structure, unfolds, becomes inactive, in the worst case scenario, that's valuable amino acids lying around being unused. The worst case scenario, it's toxic. And so the cell has a normal protein degradation pathway, and over time, proteins become ubiquitinated. Ubiquitinal.
And this is only for, so this is just within HLC, right?
No, ubiquitination is a process that occurs in the cell. So
Okay.
there are eukaryotic ubiquitin ligases. ICP0 is a virally encoded, so it's a virus protein that's a ubiquitin ligase.
Okay.
So what the virus is doing here, let me steal the take-home point of the paper. ICP0 is going to ubiquitinate the proteins that are in charge of this response to infection. So the virus is using ICP0 to ward off the cellular response to kill it before it replicates. The way it does is not a subject of the paper, but we're going to talk about the proteasome. So the proteasome is this giant complex. Anybody ever heard of GROYEL, GROYES? No? Okay, so for our, giant protein complex. Just give you an idea. It's not that impressive. That's impressive. So this is at least megadalton complex. There are many different polypeptides here, but you can think of it essentially as the cell's garbage can. So it's got two different subunits, the GRO-ES subunits that open up unfolded proteins that are delivered to it because they're ubiquitinated, are go inside, the caps close, and they're chewed up into free amino acids. So the proteasome is the destination for aged proteins in the cell to be recycled into amino acids to be made into new different proteins. So here's the schematic that pops up, which is giant core, so it's double barrel, things can come in from either side, unfolded protein put in, there's your ubiquitin, that protein has been ubiquitinated. Ubiquitin is sort of the postage stamp, or the address label saying send this protein to the proteasome. It gets destroyed in the proteasome. And then there'll be others as we go along. Okay, the other thing you can do when you're reading a paper and you're lost in an alphabet soup, as I was, even getting through the abstract, I had to go look some of this stuff up. I didn't know that IFC0 was a ubiquitin ligase. I quickly found it out. Once I found it out, it made a lot of sense. But we want to know, what the hell does that mean? What do they say at the end of the their results define a pathway for nuclear innate sensing of HSV DNA. So DNA will be sensed in the nucleus by IFI-16, that's this protein, easily enough figured out, and document a mechanism by which the virus can block nuclear innate response. I'm like, okay, before I look at their data, let me flip to the back and see how they interpret it. This is their interpretation in schematic form. So if you got to the paper, you may not have gotten to this point in the future, flip to the back and start with this. So here's your enveloped, double-stranded virus that can become that can lose its envelope by virtue of endocytosis. Endocytosis into a vesicle, an endosome, followed by escape from that endosome. These are virally encoded proteins, or direct fusion with the membrane. We wind up with a icosahedral structure that's a capsid that is trafficked to the nucleus, attaches to a nuclear pore. So see, there's a proteolytic event, a cleavage to open this capsule. It's coded for by a viral protein. The DNA is transported into the nucleus, at which point IFI-16, cell-derived protein, recognizes the viral DNA and binds to it. That triggers export of IFI-16 from the nucleus via a unknown path, there's a question mark there, through a protein called STING. I don't know what it does. But winds up in IRF3 being phosphorylated. Insulin response. I think it's insulin response. Insulin response factor 3 gets phosphorylated by a kinase called TBK1, this is one of those post-translation modifications we talked about, cellular event, in the cytoplasm, outside the nucleus. That protein, now phosphorylated, that's what the T means, is transported into the nucleus, at which point it dimerizes and turns on genes. So that the response to, holy crap, there's viral DNA in my nucleus, I'm in trouble. In this case, I'm a human foreskin fibroblast. Get that out of the nucleus, trigger the signaling cascade that turns on my genes that allow me to respond and destroy the virus. And warn my neighboring foreskin fibroblasts that, hey, I'm infected. So they can turn on an immune response to the virus, or eventually turn on an immune response to the virus as well. Now, ICP0 is that ubiquitin ligase. DNA is transcribed into RNA in the nucleus. It's a DNA virus, so that's why herpes virus has to get to the nucleus. Makes its messenger RNA. There are other genes here. This is one of the first events in the virus infection. RNA is transported to the cytoplasm. There's a ribosome, which translates ICP0, which has a nuclear import signal. IPC0 is brought into the nucleus, where it has two activities. One, it binds IRF3, blocking it from binding DNA and turning on the response that's going to destroy the virus. And it also, as we'll see, is going to degrade IFI-16 by ubiquitinating it, and thus targeting IFI-16 for destruction. So this is a clever trick the virus has. I get in, and then I shut down the cell's response to, so I'm no longer a foreskin fibroblast, I'm a herpes virus. To turn off the response in two different ways. Not allow the gene, not allow the gene the response of turning on viral defense genes, but also destroy the primary defense event, which is binding of the viral DNA. Okay, that said, let's dip into the figures. Oh, and the other thing to do, by the way, is you've written lab reports, right? You need a title, you need your lab partners, in this case it's co-authors, you need your abstract, your introduction. It's a very dense thing. This is an entire field of research in less than a page. So you're not, I didn't understand it. Doubt you understand it. When you want to understand it more, you go and they'll reference some reviews that'll tell you about the, so there are articles that will survey the history of the literature and tell you more about the field itself. But you can read the last paragraph. That always tells you what it is. When you're speaking, some people think you're supposed to say, tell people what you're going to tell them, and then you tell it to them, and then you tell them what you told them. So scientific papers are written that way. The last paragraph of the introduction generally is going to be, this is what we're going to tell you. In this study, we investigated the initial activation of IRF3 in human fibroblasts in response to HSV1. So there's IRF3 getting turned on. We observed that type 1 IFN interferon expression in response to replication defective HSV1, so you're dealing with a virus that isn't going to kill its host, it's modified, as we'll see in a second, requires the accumulation of viral DNA in the nuclear compartment. Furthermore, we identified IFI-16 as a nuclear sensor of HSV infection. In addition, we described an immune evasion strategy used by IPC0, which inhibits IRF3 signaling by promoting the degradation of IFI-16 during infection. So they say in a paragraph what I took 5 or 10 minutes to tell. So, stepping through the figures. What's ISG 54? Why am I forgetting what it is?
Oh, yeah, they just do this.
Okay, right. So I now looking back, I don't know what it is either. I do know what it is. It's a gene. So this is part of the skill, right? So going to a scientific talk, reading a scientific paper is kind of like going to watch a bunch of dolphins talk to each other. It's gonna sound like, it's going to sound like that for you guys for a while, a long time, years. But eventually you figure out that the means something. Might mean something else. Maybe you don't know what it is, but you do know it's figuring out a language by immersion. So look here, relative ISG 54 mRNA expression. Okay, so without knowing what ISG 54 is, tell me what ISG 54 is.
Well, we got mRNA expressions as a gene.
Carlos, what?
What? Yeah, mRNA expression, so I mean, obviously it's going to be a gene of some sort.
Right, so ISG 54 is a gene. Given what we know about the paper, what kind of gene is it? It's at least a virus, a gene that's going to be turned on in response to the virus. Like that just is sort of, you can figure that out. Why would they have a figure of a gene that's not turned on in response to the virus? Or say, turned on in response to eating a bunch of glucose, right? That wouldn't make any sense for the paper. So, just look back. It's an interferon responsive gene. So they're going to measure mRNA as a function of infection by a virus, and that is going to be a proxy for production of interferon. MLI? What's that?
Something like infection.
Multiplicity of infection. So it's the amount of virus it takes to infect the cells at 10 times. That's the 10 times the amount. We'll see MOI of 50 in this paper as well. And then you have to look DS106 and D106 and D109 are modified HSVs. D106, so herpes has early and late genes. I believe I mentioned that at some point. The viral genes are regulated in kind and expression. Early genes are turned on early in infection, not hard. D106 is deleted for everything except ICP0. They've knocked out all early response genes except ICP0. And they list the ones that knocked out ICP4, ICP22, ICP, who cares? This is all early, do I have it right? Yes. All early genes except ICP0. And by the way, I'm making an error here. So one thing you can learn as an undergrad is when you're talking about genes, you write in lowercase, or the initial letter at least is lowercase, not necessarily all of it, and the name is italicized. And then the protein is capitalized and not italicized. So just as shorthand when you're reading scientific literature, if it's italicized, it's going to be a gene. If it's not, it's a protein, and it ought to be non-capital. This paper does not do that. That's the other thing. You could be completely wrong the way you write a paper and still get it past the readers. In any event, the distinction here is we have viruses that can infect, get their DNA in the nucleus, can't make new viruses, so they're defective for being real viruses. But this one has the one gene, this one has the other gene. So we're interested in what does this one do differently than this one. And the answer is everything except ICP causes the response, or allows response to infection. So that's mock infected, at least I think it is. And then this is, so the black is the 109, and we get, so by the way, how are we quantifying mRNA here?
This is relative, so.
Yeah, so there's a standard, but wait, what's the method?
It would be number based on molecular weight, kilodaltons and newtons.
So I'm looking for a method here.
Like a Western
Electric gel for
No. So Western blot would be for protein. There's no gel electrophoresis. It's real-time PCR.
Yeah, that's real-time.
So you take mRNA, they have primers that are specific to the gene. So they're looking at a cell lysate, but we have primers specific to ISG54, saying look at this gene only. And remember that fluorescence assay where you do rounds of PCR, so first you do a reverse transcription of the DNA, and then you have a fluorescence signal that's destroyed when the product of the PCR reaction gets to a certain, sorry, it's this way, relative to you it's this way. And the longer it takes for the fluorescence to quench, the less RNA there is. So we can quantitate based on, it's an amplification strategy that looks at specifically the ISG54 species. And then this fold is relative to a standard. It's probably uninfected cells, so that if you just put whatever the virus, you have your virus in, so there's a solvent in there, you get more or less no response. These units don't mean anything, right? Lower is less, higher is more. They are real units, but you see they don't even put the units there. And then the 109, which has everything except ICP0, allows for the innate response. The 106 does not. The difference is the presence of ICP0, also all those other genes. But ICP alone is enough to inhibit the cell's response to infection. And then B and C make an argument about these being defective viruses. We get particle forming units versus particles. In the interest of time, since I'm losing it, I'm going to skip over that. again at the same two defective viruses, 109, everything but ICP, 106 with ICP, and 106 at a 50 MOI, so a lot. This is 109, this is 106, this is a lot of 106. And what we see is these are, so interferon, mRNA, so that's the initial response, and these are, these two are interferon inducible genes, so secondarily interferon's been turned on. In the 109, we see the cells are able to respond, they turn on the mRNA. With 106, either with some or more, we see virtually an inhibition and almost complete shutdown, certainly a slowdown. So this is hours post-infection. So it's able to respond, make interferon beta and the downstream products of that, or it's not. And again, the difference is ICP 0. And then this is what? Yes, what is that? Doesn't matter. What's the important thing about interleukin 6? Actually, it doesn't matter.
Creating response to an immune response to an infection.
So what's the difference between panel D and panels A, B, and C? Say that again. In A, B, and C, but not D. Yeah, so that's the answer I'm looking for. Something happens, we're turning on the response to virus in our less disabled but not ICP 0 contained. When we put IPC 0 in there, we turn off any response to the, we're not turning off, way down, effectively off. And then interleukin 6 is a gene that's not responsive to interferon signaling. It is what is known as a, I'm sorry, I didn't hear that.
Inflammatory cytokine?
Yes, but what is important about it relative to this, and this? It's not interferon inducible, therefore it is a control. Right? Not, as a reviewer, if you saw everything but this panel, you'd be like, well, anything turns on interferon signaling. No, it doesn't. See, here's interleukin 6. Okay. TPAC, not the name of the latest rapper. It's protease inhibitor. So it's going to prevent this from happening. So a proteolytic event has to happen. Virus can attach to the nuclear envelope, nuclear membrane. Proteolysis of the virus has to happen for the DNA to get out. So these inhibitors are small molecules. They tend to not be so soluble in water. So you, when you're doing these experiments, you do a control where you just add the solvent you use to dissolve the inhibitor. That can cause, generally they're pretty toxic and they can cause cellular responses. So that's dimethyl sulfoxide. It's a very common, less polar solvent. And so in DMSO, when we infect with 109, we get a response. So DMSO doesn't do anything to our response. When we put TPAC in along with the virus, we see a drop off. So that means the interpretation there is proteolysis is necessary. So whatever it is TPAC is inhibiting prevents response, or prevents interferon expression. So we're turning off the response, or we're not turning off the response, we're preventing the response from having to happen because infection hasn't happened, or at least nuclear invasion by the DNA hasn't happened. And then MG132, not a sports car, is an inhibition of proteasome. An inhibitor of the proteasome. And so, I'm sorry. Hang on. So responsive, so interferon expression knocked down by TPAC, and then we're looking at an interferon-inducible gene transcript here. Responds to the virus, the solvent doesn't make a difference, TPAC knocks it down. MG132 does not affect expression. So, I'm sorry, does not affect expression. In other words, the proteasome is not directly involved in the response to mRNA. So we're going to come back to proteasome if we have time, in that it's degrading the protein. So the mRNA is a marker. It doesn't necessarily mean we're making more protein. It means we're making more mRNA, which may mean we're making more protein. But it's not inhibiting the proteasome that results in this response. And then, so we're looking at TBK1. This is the kinase, phosphorylated. So out in the cytoplasm, when you infect with D109, you get, and so there's an antibody that's phosphospecific here. So this is a gel that's run out. It's a band on the gel. We turn TBK1 on. In the presence of TPAC, this is the difference. This is 90% less dense than this band. So in the presence of the protease inhibitor, the virus isn't invading as well. The response is not being turned on as well. Inhibit the proteasome doesn't do anything to the response. This is tubulin. Tubulin is a protein that makes up microtubules. It's used as a control, that you're loading basically the same amount of cellular protein, but seeing differences depending on treatment. So IFI, yes, Amber.
On the last graph, there was like the line with the asterisks on it. What does that mean on the graph?
What does it mean?
Outliers.
Well, the
Nope.
one up is like an outlier, but what's the,
So this is distribution of the samples that go to make up this bar. So this is plus minus. So you have a statistical test, a statistical quality of your mean that's shown there. So they did repeats, average it was probably 3. I don't know what Price says in the paper. But this going here means something different. But a very good question, thank you. This is a sort of paper reading skill I forget I didn't know. Anybody have a guess? Why isn't it here? So it's here, it's denoting here, and it's here, denoting those two. It's here, but not here. That's exactly what it means. It means this value is statistically significantly different than this value. And let's look at that, the error bars here. So it's probably p less than 0.005, that randomly you're going to get an average, probably 0.05 is the standard. The probability you're randomly going to get points this low compared to that is very low, so it's statistically significant. This is also significantly lower. This is not significantly lower than this. That's one reason why they conclude that the proteasome is not involved in the response. This is statistically significant. So asterisk means statistical significance. By whatever statistical test they're doing, which I'm sure they describe in the methods you look at.
It's actually written right down there, with asterisk and a little text underneath it next to the P.
Yeah, I was right. Good, thank you. P less than, the probability is less than 1 in 20.
So I'll read that.
It's probably much less than 1 in 20, because that's a very different number. But if it is less than 1 in 20, then it gets an asterisk. Thank you. Okay. IFI, so We're now going to examine IFI and STING. We've looked at entry, we've looked at the response of the interferon genes. So they're using inhibitory RNA. So this is a technique by which you can physically insert, RNA into a cell that's the complement of the messenger RNA for a gene of interest. And if that RNA is expressed, the inhibitory RNA will base pair and prevent its transcription. And so, sorry, thank you. Prevent its translation, when doing anything to transcription. So you're knocking down protein levels by preventing transcription, and thus you're doing what's called a knockdown. A knockout is when the gene is eliminated from the genome. That's pretty difficult to do, it takes a long time, it's expensive. RNA knockdowns, you have, as long as you have the RNA, you can electrically, it's called electroporation, shoot it into the cell and let it do its work. So a small RNA, inhibitory RNA that's unrelated to this doesn't cause any change in the IFI-16 level. And this is plus infection, so TBK is phosphorylated when the virus is there. GAPDH is what?
Control.
Does anybody know what the actual protein is? Glyceraldehyde phosphate dehydrogenase? Does that ring any bells?
Oh, yeah.
What bell did it ring? It should ring a bell.
Respiration.
I'm sorry?
It's involved in glycolysis.
Yes, it's a glycolytic enzyme, so it's a cytoplasmic control. It should not change in response to anything they're doing. So it's called a housekeeping gene, in this case the housekeeping protein. So it's an enzyme that allows the cell to make ATP. It's not inducible, so it's a result, it should be steady. That tells you haven't screwed up in changing your protein levels here. Anyway, when IFI-16 is knocked down to the point where it's basically out, we can't see any here, TBK is not phosphorylated in response. Levels of STING, as we'll see in a minute, don't really change. I think they make an argument that's slightly different. But we see that this is another virus. That's sorry, SEV is another virus that doesn't affect IFI-16, doesn't affect STING levels, and so when we infect with 109, or I'm sorry, I'm drawing a blank here as to this. We'll move on. Sorry, infection with HSV results in a response that is mediated both by IFI and STING. So there we see a drop in response on infection with our virus, and a further drop in STING response with the virus as well. Whereas the other virus that doesn't mediate, that's not recognized by the innate response of IFI-16, Sendai virus, I believe it is, doesn't cause a change in those levels of mRNA. One of the things about a lot of the papers is when you're reading the text about the paper, it's not on the same page as the figure. Say that. Okay, IFI localizes to the nucleus. So this is a confocal micrograph of IFI-16 with an antibody with a fluorescent tag These are the nuclei of cells. This is a light or phase contrast, so you see striated cells, and then you put the two images on top of one another. What you see is your antibody to IFI only goes to the nucleus when you stain the cells with the antibody. So IFI in the nucleus. Other panel of the figure. So we can fractionate cells very easily in the cytoplasm in nuclear fractions. It's a lysis followed by centrifugation. In the mock infected, we see no ICP0 in the cytoplasm or the nucleus. So it's not turned on. That makes sense because it's a viral protein. If you haven't infected it, why would it be there? D109 doesn't have ICP0. We don't see it anywhere else either. 106 has ICP. We see some and more when we infect with some and more. Tubulin and lamin are both markers for cytoplasm. Tubulin should be in the cytoplasm. Lamin AC should be in the nucleus. Indeed, we see that. IFI-16 is present prior to infection and remains in the nucleus. Sting is in the cytoplasm. Very good. So, leptomycin B is an inhibitor of nuclear export. When we add it to cells, things can't get out of the nucleus. Phosphorylation of TBK occurs in the cytoplasm. So, if we should have stuck this in every slide. Without the IFI-induced signaling event, unclear, we don't get sting activation of TBK phosphorylation of IRF3. So we shouldn't see phosphorylation of TBK. If we inhibit nuclear export, our hypothesis would be we don't see phosphorylation of TBK. So, not infected, infected, we see phosphorylation increase. Sendai virus also causes phosphorylation of TBK, but we want to compare the untreated and the leptomycin-treated infected or not cells. The drop in response can be interpreted how? Good interpretation, that's correct. I kind of stole my thunder with this, right? If you can't get this out, this doesn't get phosphorylated. So I'm not here to attack this figure and say it's wrong. That's their evidence supporting this. I'm out of time. I'll skip the rest of it and come back to every one of those figures. We only went through about 3 of them. Contributes to the global picture of the understanding at a molecular level. We're looking at phosphorylation, transport, transcription in response to infection. They give us a better picture of the biology of a herpes virus infection. I may want to touch on this later, maybe not. We'll see you Monday. By the way, the next paper